Thank you so much, Alon. I want to start by telling you all a story about this man. This is Alexander Fleming. He was a microbiologist in the early 20th century. Now, unfortunately, Fleming came down with a cold. But being the good scientist that he was, he did exactly what I would do, swab his own cheek and plate it on a petri dish. Now, again, Fleming had the cold, so he did forget about the petri dish and left it on his desk for about 2 weeks. But when he came back, he found something very, very interesting. His petri dish had grown up the bacteria as he thought it would, but it had also grown something else. It had been contaminated with a mold. And everywhere that this mold had colonized, the bacteria refused to grow. So there was something here about this mold that was stopping this bacteria from surviving. Now, Fleming would spend the next decades trying to isolate this compound, working with scientists around the world, and ultimately create penicillin, our first antibiotic. Now, in the decades that followed, we would come to discover hundreds of antibiotics, all from different bacteria. And with every new discovery that we made, for every new antibiotic, we knew also, as Fleming knew, that in order to survive this antibiotic, that mold had to have some genetic ability to survive it. So there are antibiotic resistance genes encoded fundamentally in the same sources that we're deriving these drugs from. And so this kind of creates a problem because in the case of in the case of bacteria, what doesn't kill you truly does make you stronger. So if you're exposed to antibiotics at a low level, you can actually develop new resistances. But how does this happen? Well, it turns out bacteria have a very special property called horizontal gene transfer. They can actually bump into each other and exchange genetic material. This would be the equivalent of if you and I were to shake hands, and if it's advantageous to me, I acquire your eye color. It's a very strange thing to do for us, but it's very everyday for them. And I guess where there's a lot of bacteria that might want to shake hands. Wastewater treatment facilities. You might not know it, but wastewater treatment is a fundamentally biological process. The contaminants that we're trying to remove from wastewater are actually degraded by bacteria that we keep in the facility. And this is where I come in, because I study how resistance proliferates in these environments and how they're released into the outside environment. Now, when we take antibiotics, which you should when you're sick, if your doctor prescribes it, you don't completely metabolize it. So when you flush the toilet, you have low levels of antibiotics that you're now introducing into this treatment facility. And so now we have these bacteria, this soup of bacteria swimming around, shaking hands, exchanging genes. And this has kind of become a problem. And we've known about this for a really long time. So scientists around the world have been studying this for decades. But there's a really big problem, which is that nobody agrees on why the results are different. Everybody is seeing, sometimes it gets better, sometimes it gets worse, and we don't know why we're seeing different things. And so I wanted to look into this, and we wanted to look right here in Oregon. So as many of you know, in Oregon, we have 2 major climate zones. This is a map of the annual precipitation in Oregon, and it's clearly divided right down the Cascade Mountain Range. And so we saw this as an opportunity to explore the role that season and geography can have. So we collected samples throughout the wastewater treatment process from 17 different wastewater treatment utilities in both eastern and western Oregon. We collected information about the antibiotics present, antibiotic resistance genes present, phenotypes, and we started looking at the relationships between these and the differences between facilities. And while we don't have a lot of conclusive data yet, one thing that we have seen is that the biosolids, this solid output from these wastewater treatment facilities, tends to be higher in antibiotic resistance genes than the liquid effluent. And a big problem with this is that 25% of the biosolids that we generate in the US every year are actually applied in agricultural settings. It helps the soil, it helps fertilize the plants, but what if it's also helping spread antibiotic resistance? And so we wanted to zoom in on this and look more into this topic in particular. So we took some pots, grabbed some agricultural soil and amended half of them with biosolids. Then we planted carrot seeds and watered them as you usually would, grew the carrots, and occasionally we would collect DNA from the soil. When we looked at the DNA, we were able to see the diversity of resistance genes present. Now, unfortunately, the biosolids amendment did result in consistently higher diversity of antibiotic resistance. Now, a previous study that we did found that these also decay much faster than the non-resistant counterparts, but nevertheless, this is a slightly troubling result. So, okay, what do we do about this? What's the next step? Well, we know that we need to keep taking antibiotics. It's important for us to stay healthy, to fight infection, but we want to return back to this wastewater environment because perhaps this is our best chance of slowing this spread. If we can improve this technology and clean up our biosolids a little, maybe we can still reuse this resource. And so we need to discover better ways of retaining the antibiotic resistance in this system, and I'm working on a project that will investigate just that. Thank you so much.